We are moving to our second talk of this morning session, a paper presented by Dorit Hochbaum from Berkeley, and the topic, as you can see, is on ranking drug data and on bi-partition. So please, Dorit.
Thank you. Good morning. The topic of this presentation is to enable observations on drug performance and translate it into the effectiveness of the drugs. Now, my background is primarily algorithmic, so I hope you won't ask me tough questions about the biology part, although my co-authors are here and might be able to answer those tough questions for me. So, the main purpose in this particular project is to use high-content screening, which provides from cell images, it provides information which is a list of associated measurements. So works like that used high-content screening extracted features. So one work is Lineal and the other one is Wang and Murphy. And then in those works, they extract multiple features, as many as 135 different features. And the question, the cells are cells that have been treated by various drugs or control group that has not been treated. And there were some previous attempts at analyzing these type of drugs, but the data is pretty overwhelming in terms of size and management of these complex data. So this particular project is an application that looks at the status of the mitochondria in cells. So healthy mitochondria would be intact, and then, so this is the upper row, and in the bottom row, these are cells that have been treated with squamotrin, which causes the mitochondria to be fragmented. The other cases were also treated with squamotrin, and in this case, we also applied two different drugs for the purpose of this study. So one is ZIETD, and the other one is ZLEHD. And these are cases that are not obvious which way they go, whether they are considered fragmented or whether they are considered intact. So we like to evaluate for each cell that was that was treated with particular drug, how close it is to being intact. Now, one attempt was to project the images. This is the corresponding feature vectors, to project them in 2 dimensions. And the picture that is presented here shows that those feature vectors tend to overlap a great deal and the cell properties cannot be distinguished by looking at the feature vectors alone. The idea proposed here has its base in image segmentation, which is an area in which I worked quite a bit in the last 5 years, and clustering. In image segmentation, a typical issue is to take an image and to extract, to segment the salient feature in the image. So there is something that in mathematical terms or statistical terms you could call a cluster, something that is cohesive in some sense. And in that regard, what happens in segmentation is that we get the feature and we have the background. So we have a bi-partition into 2 parts. So the idea was to apply types of bipartition algorithms that have been successful in image segmentation in this context. But first, the issue here of ranking is not exactly a bipartition. The main idea of this fractional adjusted bipartition score is to convert information that comes from bipartition into scalar scores. So the prototype approach is to apply a bipartition algorithm, let's call it A, that separates the more intact cells. So we will look to cluster the better cells, the more intact cells, in a way that is right now not entirely quantifiable, from the more fragmented cell images. And the score will be a number that will be simply the fraction of number of cells that were treated in a given with a given drug that fall into the more intact portion. So in the same way that in image segmentation we have the feature, here the feature, the cluster of the feature will be the more intact cells. And one application that we used is support vector machine. Support vector machine can be used, is very famous data mining algorithm, and we used it as a bipartition in this application, in this setup. So the general structure is, first of all, to construct a graph, which I'll tell you shortly how this is done. Once the graph is constructed, we apply the bipartition algorithm, whatever it is. We'll apply several. And then we recover the scalar score for each drug, because we know in advance which drug population. The algorithm itself is blind to the association of each cell image with a particular drug. But once the bipartition is achieved, then we look at those labels and we assign higher score to those that have higher percentage falling in the more intact set. So here is how the graph is constructed. We have, say, n high content screening images. Each one corresponds to a node in the graph. And each one has an associated feature vector, which, as I mentioned, would be very large, high-dimensional vector. Then we have k-population sets. The population sets are, each population set corresponds to the application of a given drug or a control group that has not been treated at all. Then we have so-called two training sets. So the algorithm needs to know, to get some information about what's considered intact and what's considered fragmented. So for this purpose, we have training sets that we call R1 and R2 that represent those extreme sets. So R1 is a bunch of images of intact mitochondria cells, and R2 is a bunch of images with totally fragmented mitochondria cells. So the graph consists of the set of nodes, the set of edges, those labels that correspond to the population set, and set of weights, which I'll tell you now how they are achieved. So basically, we are looking in terms of set of edges at a complete graph. So that is, for every two nodes, we have some statement on the similarity between them. And each node has a label that corresponds to the population set that will be either a drug or control set. And the weight corresponds to each pair. And for each pair of nodes, let's say ij, it computes similarity strength. Now this computation, this is called exponential or Gaussian weight. This is very common in image segmentation. Of course, anything else could be introduced here as a function that measures the similarity. But here, this particular function, what it is, it's exponent to the minus the difference in Euclidean space, the distance in the Euclidean space between the two feature vectors of I and j. So that accounts for the fi minus fj. And then we add some adjusted of epsilon to, because in many cases this will be pretty large, a pretty large negative quantity. So that will be very close to 0, so we added epsilon. And there are other possibilities of adjustment, but in the study, this is the type of function that we used for similarity. And as I pointed out, this is very common in image segmentation, where instead of the feature vectors, they have the color intensity of each pixel. Now, in the scoring algorithm, we can use as a black box any bipartition algorithm. It only needs to satisfy the positive control set points, so these are all the intact cells, are included in the set. And the negative control, these are the training sets that should not be in the set, are outside. So that's the only requirement, which is typically in most algorithms is very easy to include as training sets. So to repeat, R1 and R2 are the extreme cases where the R1 is the intact, the positive controls, and the R2 are the completely segmented, the negative control. And we'll want to find drugs that have scores that puts them closer to R1, that is, as many of their sample cells are in R1. And that will be considered more effective. So here is a formalism for the algorithm. So we have the input, the graph that was created, and then we use a bipartition algorithm so that that separates the graph into a set S and its complement, so that R1 belongs to S and R2 belong to the complement of S. Then for each population set Pi, we calculate the FABS score, so that the fractional adjusted bipartition score, which will be the number that fall in S of this population I, divided by the total number. So this will be a number between 0 and 1 associated with each population set I. In step 3, we sort those scores, and we get a permutation pi 1, pi 2, up to pi k, so that pi 1 is the most effective drug, pi 2 is the second most effective, and so on. So a simple example here, you have the green nodes here are the R1, the blue nodes are the R2, and then we have 3 populations, A, B, and C. And then we get the bipartition that is described by this line. And in this bipartition, you see that of the nodes of A, of the instances of population A, there are 3 instances. All 3 of them fall in the desirable set. So the score is 1, 3 out of 3. For B, 1 out of 3 falls in the desirable set, so the score is 1 out of 3. And for C, none of them fall, so it's 0 out of 3. So the outcome in this particular case will be a sorting that drug A is more effective than drug B, more effective than drug C. Now, there are lots of bipartition algorithms. So these are the ones that we chose mainly because in previous works I chose them in image segmentation and pattern recognition algorithms, and they proved to be effective. So one of them is support vector machine, and there are some specification on the particular parameters that we chose. So C equal to 10 to the power 4, and the kernel parameter is 1. The paper gives the details of the particular kernels that we used in the support vector machine implementation. Another algorithm that has been successful in image segmentation is called Normalized Cart prime, and I'll give you some detail on that a bit later. Another technique that is very common is the eigenvector technique or spectral technique. In this technique, again, it's borrowing from image segmentation. The associated graph, where one creates similarity weights, one builds from it a Laplacian matrix and then finds an eigenvector of the Laplacian matrix, and the Laplacian matrix, this eigenvector that's called the Fiedler eigenvector, it's the second, corresponds to the second smallest eigenvalue, is a very popular technique in image segmentation for the purpose of clustering and feature in an image. So because of its popularity in this field, and actually beyond this field, there has been a lot of work done on this so-called spectral technique and analyzing the properties of the eigenvector, particularly if you heard about it, there is Chigger constant and there is the expander of a graph, which are hard problems, and the spectral technique was used as a heuristic that has been extensively analyzed for close to 50 years now. So that's the reason it was selected. I'll elaborate some more on this normalized cart prime because most people did not hear about it. It is, the way it is employed here is that we look at the following optimization, an optimization criterion that involves two parts. The first part is to separate as much as possible. So CS S bar is the total similarity between the set S and the set S bar, and we want this similarity to be as small as possible. This is also called a cut. So if the total weight of edges that have one endpoint in S and the other endpoint in S bar, that's the notion of the cut. And then we also want to maximize the total similarity within the set S. We want the set S of the intact one to be in some sense homogeneous. So this goes into the objective function as well. Originally, this problem was given as a ratio problem, but we found that it's more effective to use it as essentially a weighted combination of these two objectives, and beta is a coefficient that will be selected as tuning parameter to trade off this intra-cluster similarity versus the inter-cluster similarity. So this is additional notation, but given that I have only 5 minutes, perhaps I'll skip ahead and I'll only tell you that there is a similarly looking objective function that's called Normalized Cart. It was introduced by Shi and Malik in 2001 and has been used extensively, not only in image segmentation, primarily in image segmentation, but also in biological applications. And this is to minimize the cut divided by the sum of weights of nodes inside the set S, plus the cut divided by the sum of this corresponding weight of nodes in the outside set. And this problem is NP hard, whereas the problem that I showed you is actually polynomial time solvable. So that makes it very effective. Okay, this is the formulation, and I'll skip that. And I'll just tell you that we chose several other ranking algorithms that were used. So one of them is center ranking, which is very obvious and quick. You find the median of the feature vectors in each population set. The other one is to append to the center ranking the principal component analysis. And a third one that in a paper that was published by Lineal in 2010 is called graph transition energy method. And this one is time consuming. So let me show you quickly the results. So these results are in terms of accuracy, that we get a ranking of drugs and we know the ranking ahead of time. So this is a simulation. And every case, so we ran it 1,000 times, and every case that in which the score, the ranking order coincided with the correct permutation counts as 1 towards the accuracy. So the percentage of the total is marked here. And this is the Fab's NC prime. So this normalized cut prime is the higher one, and the horizontal axis is the training sample size. So it becomes more and more accurate, and the others also, in general, become more accurate, although not entirely. Some of them are going up and down. It becomes more accurate as the size of the training set, a percentage of the training set increases. So it pretty much dominates the other ones in terms of accuracy. Now, in terms of speed, it is, for the smaller training sets, it is runs less efficiently than the one with support vector machine. But for the higher training set, it runs more efficiently. The other, the GTEM is the blue one that has the worst running time. And the center ranking, as I said, it's a very quick procedure, just finding the mean of the feature vector. That, of course, is very quick and not affected by the size of the set. So overall, although you put some more running time than the trivial case, the running time is very good, and you get substantially higher accuracy. And these are some comments about future research that we just started to look at peroxisomal data. And this is a chart that shows the whale numbers, and the whales in this chart are organized, so these were negative and the other ones were positive, and as you see, there is a clear distinction between the two sets that was achieved with the Fab's NC prime approach. So I'll just thank you and take questions. Thanks.
Thank you. Dorit, please, can you present yourself also?
Sure. So thank you very much for the talk. I had a question about the normalized cut objective function. I was curious if it's...
But you didn't present yourself.
Oh, I'm Corbin Rivera from DuPont. And I had a question about the symmetry in the normalized cut objective.
Right.
So I noticed there's a term that maximizes the within cluster distance for S, but I didn't notice a complementary term for S bar. I was just curious if you could comment.
That's, you just put your finger on it. That's exactly the source of the difficulty. This problem is the normalized cut is NP hard. And in a way, it wants to partition into two sets that each one is a homogeneous cluster. And that makes it very hard, that you want both of them to behave nicely. But in many image segmentation applications, you just care about the one set, the background, you don't care about the background. And taking this philosophy in this case, you might say that you do care about the background, which is the fragmented cells in this case. But the price you pay for it is very high because the problem becomes NP hard. And in fact, this, what I call NC prime, is also a very good approximation to the actual value of this objective function. That is, I get a partition. If I plug it in, I get a better objective function value in general, in practice, than the spectral method would provide. So it's pretty good also for that. Thank you.
Please.
Hi. Hello. Hi, I'm Saad Sheikh from University of Florida. I wanted to ask, do you know if your new objective function has multiple optimal solutions? Any ideas?
No, you solve it optimally. I mean, there are rare cases in which all of them are of the same value. But in general, because the similarity weights are essentially real numbers, then it's pretty rare. I mean, we haven't come across any instance where there are multiple optimal solutions.
Okay, thank you.
Mm-hmm.
Thank you. And maybe if I can just comment on, I promise not to ask any hard question on biology, but there is a very nice resource in the Human Proteome Atlas from the Sweden European, and they have hundreds of images in which they are manually annotated as peroxisome, not peroxisome, and so on. So maybe it could be a test case because they come from a really very careful annotations team.
Yeah. The thing is whether they'll have also associated feature vectors, because this technique does not really look at the images. It looks at the extracted feature vectors and analyzes them. It doesn't look directly at the images. There has to be an intermediate step.
Yeah, it comes with a very, very detailed images, so you can take the images before even, the raw data, which is very high quality.
Great. Thank you.
Sure. Okay, let's thanks Dorit again.